Welcome to the Deep Dive. Today, we're giving you the ultimate shortcut to understanding human strength. Our listener is looking for, well, the scientific blueprint of movement, and we've compiled a stack of sources that takes us from the weights you lift.
Right down to the molecules that power them.
And that's really our mission today, isn't it?
It is. We are unifying the macro, the types of movement you actually perform in the gym, with the micro, the incredible molecular and energy systems that make all of that possible.
So by the time we're done, you won't just be doing a workout. You'll understand exactly how your muscles are recruiting energy, why one type of contraction stimulates more growth than another,
And how your body decides which fuel tank to tap into.
It's the difference between just doing the exercise and, commanding your own biology.
Ready to dive in.
Okay, let's unpack this. We have to start at the foundation. The three fundamental ways a muscle can generate tension. So, when we talk about generating force, everything really boils down to one simple question.
Is the muscle length change or not?
Exactly. And the first of our three classic modes, is one everyone recognizes, but it technically performs no mechanical work. I'm talking about isometric contraction.
Isometric. So iso means equal, and metric means length. Equal length.
Right.
The muscle is developing this significant force, a lot of tension, but its overall length and the joint angle, they just stay static. Think of pushing against a wall.
You're trying your hardest, but the wall isn't going anywhere.
Exactly.
Or the dreaded plank. I think we all know that feeling. You feel that immense burn, that internal tension is just building and building. But since the distance traveled is zero.
Mathematically, you've done zero mechanical work. It's purely about stability, static endurance.
Okay, so that's if nothing moves. What if the muscle length does change?
Then we move into the dynamic categories. We call them the isotonic contractions, and this is split into two types, concentric and eccentric.
Concentrics is the classic one.
Yeah.
Actual lift.
That's the one. If you're lifting a water bottle, pulling yourself up on a pull-up bar, the muscle shortens. The force it generates overcomes the external load. It's the power stroke.
To the upward movement of a bicep curl. You're fighting gravity.
You're fighting gravity and you're winning. That phase is, well, it's traditionally associated with movement initiation and hypertrophy.
Okay, so then you have the third type, eccentric.
Right, the controlled lowering. The muscle is actually lengthening while it's still resisting that external load.
So the load is winning, but you're managing its descent, like walking downstairs.
Exactly. And here's where it gets really interesting. And it's a point a lot of people, even trainers, miss. Contrary to focusing all your attention on the lift, our sources show that this controlled eccentric phase, it actually leads to greater muscle stimulus.
Wait, greater than the concentric part?
Often, yes, and greater maximal force gains.
That feels counterintuitive. Why is the controlled yielding, the negative part of the lift, a stronger trigger for growth than the actual lifting part?
It all comes down to leverage and recruitment efficiency. When a muscle is actively lengthening while resisting a heavy load, it actually requires fewer motor units to produce the same force output.
Fewer.
So because fewer motor units are needed to handle that same load, the individual muscle fibers that are recruited.
They're placed under a far higher mechanical tension.
Precisely. That elevated mechanical tension plus the micro trauma that results from it is a really powerful biological signal for adaptation. It just maximizes growth and strength.
Which is why trainers are always yelling about slow, controlled negatives.
That's exactly why.
Okay, so we understand the movement now. Shortening, lengthening, or just staying still. But how? How does the muscle physically generate that tension at the molecular level? We need to get inside the engine.
Right. So if the muscle is this complex machine, its basic tiny contractile unit is called the sarcomere.
The sarcomere.
You can picture it like a zipper. It's defined by two lines on either end, the Z lines. And within that zipper are two main proteins, actin.
The thin filaments.
And myosin, the thick filaments. And the fundamental action of all strength, what we call the sliding filament theory, is simply when these two filaments slide past each other.
So they just slide. They don't shrink or anything.
Exactly. Imagine the myosin filaments are like 1,000 tiny oars, and the actin filaments are the rope they're pulling. When the muscle contracts, the Z lines move closer together.
Because the oars are pulling the rope toward the center.
Perfect. The filaments themselves don't change length at all. They just overlap more.
That makes the structure clear. But what tells those thousands of tiny oars to start rowing all at once?
That requires an electrical signal, a nerve impulse. It travels across the muscle cell membrane, the sarcolemma, and then it dives deep inside the fiber.
How does it get deep inside?
Through these specialized tubes, the T-tubules, think of them as the electrical wiring that runs the signal deep into every single corner of the muscle cell.
And that wiring, it triggers the release of the real key player here, calcium.
Correct. The T-tubules connect right to the cell's internal storage battery for calcium, the cercoplasmic reticulum. When that signal hits, this reticulum just floods the interior of the muscle fiber with calcium ions.
So what happens when that calcium flood begins?
The calcium binds to a regulatory protein called troponin, which is sitting on the actin filament.
Okay.
And that binding causes a structural shift. It essentially pulls a protective cover, another protein called tropomyosin, right out of the way.
So tropomyosin is like a safety lock.
Yeah.
And calcium is the key that removes it.
That's a great way to put it. It removes the safety, exposing the binding sites on the actin. Now the myosin ores can finally grab onto the rope.
And that initiates the cross-bridge cycle.
Yes. The myosin heads, which are already in this high-energy cocked position, they attach to the exposed actin sites. Then they tilt forward, that's the power stroke pulling the actin filament, and that pull is what shortens the sarcomere and generates force.
Okay, this sounds like it must require a ton of energy. Which brings us to the critical fuel here, ATP.
It does. And here is probably the most surprising fact about this entire process. You need ATP, adenosine triphosphate, not just to energize the myosin head for the power stroke, but most crucially, a new ATP molecule has to bind to the myosin head for it to detach from the actin.
Wait. ATP is needed for the release, not just the pull.
For the release. If ATP is unavailable, if your body is completely out of fuel, that cross bridge can't release. The muscle just stays locked in a contracted state.
And that's the chemical explanation for the stiffening that happens after death. Rigor mortis.
That's it. The motor runs out of fuel and just jams.
Okay, so if we connect this to the bigger picture, all that attaching, detaching, and pulling requires a constant supply of ATP.
An immediate and continuous supply. And the body doesn't just rely on one supplier. It has 3 major fuel systems that kick in based purely on the intensity and duration of your effort.
It's like having 3 different cars for 3 different trips. You've got a lightning-fast sprint car, a strong mid-range vehicle, and an endurance tanker.
A perfect analogy. Let's start with the fastest one. Immediate power.
The phosphagen system.
Our speed demon, it lasts for only about 8 to 10 seconds of maximal effort. We're talking a 100-meter sprint, a vertical jump, a single max effort lift.
And it uses the tiny bit of ATP that's already just floating around, which is gone in, what, 3 seconds?
Less than 3 seconds. And then it relies almost entirely on something called phosphocreatine, or PCR.
Right.
PCR is the genius of the system. It's in the muscle fibers at concentrations way higher than ATP, and it acts as this emergency battery. It rapidly recycles used ADP back into fresh ATP. It's explosive, but very short-lived.
So once that PCR emergency battery is drained, we move on to the second fuel tank.
We do. The mid-distance powerhouse. Anaerobic glycolysis.
Anaerobic, meaning without oxygen.
Without oxygen. This system takes over from about 10 seconds up to around 90 seconds, maybe a bit more. It breaks down stored glycogen or glucose for energy. It's still really fast producing ATP, about 2.5 times faster than the slow aerobic system.
Perfect for something like a 400-meter run then.
A 400-meter run. A long wrestling match, yeah. Now, the famous byproduct here is lactic acid, which everyone for years blamed for muscle fatigue.
The burn.
The burn. And that's a misconception our sources really clarify. Lactic acid is not just metabolic waste, it's actually a crucial source of fuel.
Fuel for what?
During intense exercise, your heart muscle is working incredibly hard, right?
Yeah.
Well, it actually uses lactate as a preferred fuel source. The body is recycling its own waste in real time.
So for all those years we blamed the burn on this acid, our own heart was sipping on it like an energy drink.
That's a great way to think about it. It just speaks to the body's precision. In fact, that precision is best seen in how the body controls this whole process with an enzyme called PFK.
Phosphofructokinase.
Yeah.
The rate-limiting step.
Exactly. High levels of existing ATP actually inhibit PFK. The body sees it has enough fuel and it hits the brakes. But when energy is low, marked by high levels of ADP or AMP, those molecules strongly activate PFK.
So it instantaneously matches fuel supply to demand.
Perfect.
Okay, and finally, we reach the biggest tank of all, the marathoner, aerobic oxidation.
This is the system that uses oxygen to break down carbs and, eventually, fats. Its ATP production rate is the slowest, but the yield is just massive.
Wow, massive.
Up to 38 ATP per glucose molecule. Its duration is virtually unlimited as long as you have fuel. It powers everything from jogging to long hikes.
And these different energy systems are physically embodied in our different muscle fiber types, right?
Absolutely. The slow twitch fibers, type I, are packed with mitochondria. They're red because of all the myoglobin. They're specialists in this aerobic system.
Fatigue resistant.
Very fatigue resistant, found in postural muscles. On the other hand, the fast twitch fibers, type IAB, are white. They're optimized for those anaerobic systems and provide that explosive short burst power you need for sprinting.
We've covered the basic movements and the core science. But to really apply this knowledge, we have to look at two more contraction types that kind of bridge the gap between theory and the real world.
Definitely. And the first one is actually the one you use most of the time. It's called oxytonic contraction.
Oxotonic.
If you lift free weights, this is what you're doing. Unlike the theoretical isotonic ideal where tension is constant, oxytonic acknowledges that both the muscle tension and the muscle length are changing continuously through the movement.
Because of gravity and changing leverage, right?
Yeah.
The weight feels heavier at different points in the lift.
Precisely. The resistance is always fluctuating.
Okay, so that's real life. Then we have the precision tool, isokinetic contraction.
This is a highly specialized dynamic contraction where the key is that the speed of movement is kept constant.
Not the resistance, the speed.
The speed. The angular velocity is kept constant by a special machine, an isokinetic dynamometer.
How does that work?
The machine automatically provides variable resistance that exactly matches the maximum force your muscle can generate at every single point in the range of motion.
So your muscle is maximally loaded through the entire movement, even at its weakest angles.
Which is impossible with free weights.
This raises an important question then. Why is that controlled, constant speed so critical for, say, rehabilitation after an injury?
Safety. It's all about the inherent safety mechanism. Since the machine's resistance is always equal to your effort, if you get tired or if you feel pain and back off even a little,
The machine instantly reduces the resistance to mat.
exactly. It prevents you from ever overloading an injured joint. It's the safest way to maximize strength development across the entire range of motion post-injury.
It also sounds like the most objective way to actually measure strength.
It absolutely is. Isokinetic testing is the gold standard for measuring functional performance. Clinicians use it to identify crucial strength imbalances, like bilateral asymmetry.
So a difference between your right and left side.
Right. If that force difference is more than about 10 to 15%, that's when injury risk really starts to spike. They also use it to confirm ratios, like the hamstring to quadriceps ratio, before clearing an athlete to return to play.
We started this deep dive seeing simple movements, like a wall sit or a bicep curl. Now we know those actions are supported by 3 different fuel tanks.
Immediate mid-range and endurance.
And they're powered by these molecular engines that require ATP not just to pull the muscle shorter, but to physically detach the proteins.
That level of microscopic control is just so precise. The fact that the body regulates key enzymes like PFK based on whether it sees high-energy ATP or low-energy AMP, it shows how perfectly our metabolic demand is matched to our physical output. It's a beautifully precise system.
Which leaves us with one final provocative thought for you, the learner. You now know that your sheer strength isn't just about the size of your muscle fibers. It's about the precision of the motor units your nervous system decides to recruit. Next time you perform a movement, remember, Henneman's size principle governs every single effort. The nervous system always starts by firing the smallest, most precise motor units first. It only graduates to the massive, powerful units in your glutes when maximal effort is absolutely required. So every movement you make, from texting to squatting, is actually governed by the exact same neural rulebook. Keep learning, keep moving.